I'm Fiona Greer, and I'd like to welcome you to the fourth in our series of short, informal roundtable discussions on the theme of biosimilar analytics, hosted by BioPharmaSpec. Today, I'm joined again by two experts who sparked such a lively talk last session on knowledge-based biologics development that we had to split it into two. So please look on the website to see that discussion. Welcome back, Dr. Andrew Reason and Dr. Ash Ramzan. They are keen to start getting their teeth into the topic of when and how should biosimilar analytical methods be validated during the development process. Andrew is founder, CEO and MD of BioPharmaSpec. He has 25 years of experience in the analysis of both novel and biosimilar biopharmaceuticals, and has been involved in the commercialization of a number of analytical methods for characterizing proteins. Andrew has contributed to many industry publications and is a regular presenter at conferences. He is also currently a visiting professor at the University of Warwick and recently joined the Macquarie University Scientific Advisory Board. Ash is founder and principal consultant of Woodley Bioreg, a global regulatory consultancy. He has a specialist interest in biologics, biosimilars and vaccines, and is a member of the British Pharmacopoeia Working Party, advising the government on the development of biosimilar standards. With over 30 years of hands-on expertise, Ash continues to work extensively in developing regulatory strategies for global registration of biologics and biosimilars. Ash is also a fellow of TOPRA, as well as serving on their editorial board. So thank you both for joining me again today. Now, a question I'm perennially asked is, when should I validate my methods? So I'd love to hear your take on this during our discussion. So before we start off, just to make things clear, is qualification and validation of analytical methods the same thing in your eyes? Ash?
I guess it's worth it's a difficult question. It sounds very obvious, but it's actually difficult to answer because aggressive development timelines for biopharmaceuticals means that that distinction starts to become clouded as applicant sponsors move rapidly through clinical development. So the strict regulatory view is that by the time you perform your phase 3 clinical studies, everything should be validated. So methods, cleaning, process, facilities, utilities, everything has to be fully validated so you can use that status for your ongoing clinical studies and reporting. However, that assumes that this is a binary process that goes through. It doesn't. Methods are being developed, optimized, qualified, validated on an ongoing basis. So it's not unusual to see manufacturers that have gotten to phase 2, sometimes late phase 1, and have got essentially validated tests, and oftentimes in other instances have reached phase 3 and the test would only, by the purist view, be qualified. So our view is, again, are the tests fit for purpose? Have you looked at those parameters that potentially could be validated, that could qualify or validate, improve, or otherwise enable the interpretation of data? There's a grey area there. My view is that regulators, if you can discuss this with them during scientific advice, will accept your position, but they will never do so in writing, and they will never do so in a regulatory framework. But they understand the real life situation around these types of processes. Okay. I haven't answered your question, Fiona. I'm sorry, because it's quite a difficult one to answer.
Yes, it's a difficult question to answer. It's not a yes/no answer, is it? It's a depends answer, isn't it? So, well, Andrew, what is an analyst perspective then? We know that the method should be fit for purpose, appropriate, and applicable. From your expertise, what would you say about that?
Well, yeah, some of it echoes what Ash is saying, really. I mean, I think if you lined up 10 analysts, they'd all give you a different answer, and that's part of the issue. I mean, what we do at BioPharmaSpec means there is no real distinction apart from one thing. You know, a true validation when you're validating methods for release tests has to be done to GMP. But all validations or qualifications should be carried out to the ICHQ2 guidelines. So at BioPharmaSpec, because we work in the early phase development, we're not in the clinical phase. We call all validations or qualifications. They're done using all the parameters from ICHQ2, and you know, looking at analytical quality by design, and I think we talked about that a bit in the last discussion. The more you know about your method before you actually do the qualification, the better off you are, and you have a strong package that then leads into the true validation phase, which you know is in the clinical phases, as Ash mentioned. So I think there's one other important point that we have talked about before as well, and that's, you know, qualification per se. If you look at the requirements for a qualitative method qualification, it's all about specificity, which is fine. You know, you can look at interferences and all sorts of things, but if you want to understand the variance of that method, specificity isn't going to tell you much about that. So for all methods, irrespective whether it's qualitative or quantitative, we would always look at repeatability. We would always look at intermediate precision. We would look at different operators, different days, you know, different columns, if it's an LC method and everything else, to get a very good grounding during development. Qualify the method as if it was a GMP validation, and then you're at a very strong point, you know, whether it be using standards of proteins, peptides, or oligosaccharides if it's a non-critical method, or whether you're qualifying with the product if it is a critical method. You're in a very strong position as you go through the development phase into the clinical phase.
Well, following on from those comments then, Andrew, what about the recent revision of the ICH validation guidelines, the Q2R2? Will this make any difference to your approach?
Not to ours, but there are a few things in it that have more focus, shall we say, than they did before. Robustness is one of them. You know, showing your method is reliable and robust. There's a lot more information in there, and you know, I would say to anybody, go out there and look at the draft and keep an eye out for the final revision 2 document. There's a lot of information in there about how you report residuals and that sort of thing, and more information being required on how you define LODs, LOQs, that sort of thing. But you know, that, to be honest, they're the things you should have been doing anyway. They've just risen in terms of the priority and the focus of the document, rather than there being anything new. But I think, you know, if you consider Q2R2 and you consider the analytical quality by design information that's coming out, the method development phase is really the most important, to understand your method in depth, such that, you know, the results of the qualification are pretty much already known, but it's being used to get that information on variance, look at experimental error, and be able to say whether the result is comparable to another one or not.
Ash, do you feel that these revisions and things are coming out as a consequence of regulatory comments?
I think the regulators, to a certain extent, are also acknowledging and recognising the limitations in terms of the accelerated development of complex biological molecules. And I think Andrew's described a really conscientious and diligent approach to method development. Not everybody is the same, but what Andrew, you mentioned was the development phase challenges the test in areas that you wouldn't normally expect to challenge your test. And I think there's that lends itself to this whole argument of, where does development stop, optimization start? Where does optimization stop, qualification start? Where does qualification stop and validation start? Well, we don't know, because it is a continuum. So you may well be using data that you had in your development or optimization phase that you use then in validation. And I think during one of the earlier sessions, we talked about being fit for purpose. That's exactly it. Any test that you apply has to be fit for purpose at the time that you're looking to apply it, to the molecule that you're looking to apply it for, and for the purpose you're looking to apply it for. So all of these parameters play in, and I think the regulators are trying to grapple with how do you put that into is it a regulation, is it a guidance document? How do you get industry on board with that? And we always take the position that there are going to be so many areas within this CMC realm that are not fully clearly defined. So we're happy to go to the regulators and have a discussion with them, knowing that we're not going to expect answers, but we're going to get a steer as to what their expectations might be, knowing also that they've seen similar problems, similar challenges with similar products from other manufacturers.
Another question which is often asked, do methods need to be qualified using the actual drug product, or are qualifications using standard proteins, glycoproteins, oligosaccharides, etc. sufficient? Andrew, from an analyst's point of view.
Yeah, I think that the answer really comes from our experience with the regulators, and I think methods that are supportive, i.e. they're not critical in terms of decisions made about the product as it moves through the development pipeline. The EMA and the US FDA in our experience are accepting that methods can be qualified with standard proteins, peptides, saccharides, that sort of thing. So you have a good idea about experimental error. And when you're comparing, a new batch to a reference batch for a new biological or an innovator to a biosimilar in a biosimilar study, you can say whether something is comparable or not within the expected experimental error. Where product development and product qualification comes in is for those methods that are fairly critical and really look at the critical quality attributes of that particular product, or are being used by the company to make those decisions to progress through the development and into the clinical phase. I mean, as you develop into the tests that are going to be perhaps the GMP release tests, then those are often the methods that we would be qualifying at an earlier stage using the actual product on top of the qualification that we've done using standard peptides, proteins, or whatever it might be. So it's a development. There has to be discussion between ourselves and the client and the regulator, and it's never the same twice. Basically, the products will dictate certain methods, the development and the knowledge and the data you're producing in those early phases will, you learn a lot about the product and which methods are critical to assessing that product. And generally those are the ones where you would be carrying out a product-based qualification, which can then be quite nicely transferred into the GMP environment as the basis of a GMP validation prior to creating the release test.
Ash, do you wish to comment on that?
Yes, I could just add maybe a couple of points. I agree that the tests, sometimes you can use surrogates to do the latter stage qualification, early validation, but there are going to be some instances where you just can't do that. So if you think about, so ultimately what you want to be at, so the regulator will want to know what is it that the patient receives. Well, it may be a product that's adjuvanted. You can't strip it off you can't strip a vaccine off an adjuvant, so that's impossible. It may be a toxin that's administered in nanogram quantities. Well, you can't analyze that. So where there is a justifiable reason to move upstream to a representative drug substance element, a bulk drug substance, that's always acceptable to a regulator, but you have to define why that's important. So I agree with what Andrew said, but actually I would add in a couple of other examples of where it's actually almost impossible to analyze, characterize the final product.
Yeah, blood circulating glycohormones is certainly an example of that, where the drug substance fairly late on might be around 1.6 mg per ml, but the formulation is around 50 micrograms per ml. So yeah, Ash is correct. Yeah.
So we've discussed a lot about the continuum of this development and the fact that we do often loop back to earlier information in our knowledge-based development. But as a final question, in your opinion, at what point in the product development pipeline is this initial structural characterization and any required qualification complete? I don't know whether you can give a response to this, but it's just the question that I'm also always asked, and I'm sure you're always asked, when should I validate my methods? Or is it really on a case-by-case basis? There isn't an answer to that question. Andrew, do you want to give your view on that?
Yeah. I mean, it is a tough one. I mean, it depends on so many things. You know, the method development in our hands needs, you know, a decent number of batches. You need to analyze a decent number of batches to get that core structural information, understand something about variability and how your methods relate to that product. You know, if you're just producing one huge batch and trying to do everything, it's way more difficult to do the method development and look at different parameters than if you're producing several and can relate all that data and look at, you know, scale up or change in site or whatever it might be. So, you know, I think with every product, and it's usually, you know, at the end of the development phase where you have a good amount of data, or you have all the data you're ever going to get. Do you know what I mean? And maybe it's not enough, but you kind of have to know enough, obviously, about which methods are going to be critical to that product and which are just going to be supportive. Yeah, and that often comes with initial discussion with people like Ash or the regulator or whatever it might be. And then, you know, you're at a point there where you probably are wanting to qualify those methods to understand the experimental variation and what you feel about comparability and whether something is similar or not is actually true. There are exceptions to everything. You know, the one thing that's very important with a biopharmaceutical sample is how much of it are you actually putting into that syringe and into that clinical trial. And ICHQ-6b says that, you know, the gold standard for doing that is amino acid analysis with optical density measurement and creating an extinction coefficient. Now, that is a pretty important cornerstone of any product development, and we always qualify that. You know, that is something we always recommend is qualified, and we do that pretty early on once a product is, you know, has the purity that the client wants, and obviously quite importantly is in the formulation that is probably going to move forward, although that can change, but you can always go back and reassess and make sure nothing has changed and all these sort of things. So, you know, it's, you know, it is a continuum, which is a word Ash has used through some of these discussions, and, you know, it's happening real time, and, you know, it's not as if you're marching towards a point where you qualify everything. You're qualifying things at different stages, depending on the importance and depending on how many batches you've analysed and what data is coming out of that. But certainly we would be directing clients to have all the important critical methods or methods that assess critical parameters qualified towards the end of the development phase. And that's the simplest, I guess, answer I can give.
All right. Ash, your final comments on this continuum of development and when should the methods be validated? Well,
I think I can just add maybe one or two very quick points at the end there, because Andrew, you've covered a lot, and I think you're right. But Fiona, to answer your early questions, it ever end? No, because these analysts keep developing new methods, new ways of interpreting data, and it's incumbent on developers to go back and look at their products. But in terms of when do you validate, from a commercial product development standpoint, you want to try and have something that's as near complete as possible, not necessarily fully validated, as early as possible in the clinical development phase. So we work with clients where they seek to fully qualify, pseudo-validate their products by phase 2. What that means for them is they can use the phase 2 clinical data in phase 3 reporting, and that accelerates their clinical development. So in and amongst everything else that the developers are doing, the analysts are doing, the regulatory people are doing, the commercial people are trying to shorten the development pathway to a point to enable phase 2 data to be used within phase 3 studies. We do see that, but it has to be discussed, negotiated and agreed with the regulator, because they need to understand that a full data validation package isn't necessarily going to be available at that time. But then you come back to the academic question of what is full validation, because the goalposts keep changing.
Well, on that, we might have a topic for a further discussion. I might come back to you with some questions on that. But again, we've exceeded our allocated time, so we're going to have to cut us off again now. I'm sorry about that. I want to thank you both for what's been a very insightful discussion. Thank you for sharing your opinions. Thank you all for listening, and to BioPharmaSpec for hosting. If you missed any part of this conversation, the previous conversation with Andrew and Ash, or the very first roundtable that we hosted, which involved 3 different expert speakers, then please look on the BioPharmaSpec website and look out for our next event by visiting their web page and following on LinkedIn. And I'm sure we'll have another interesting discussion with Andrew and Ash in the future. Thank you all again.